Hello everyone, welcome back to our Plante. In today's lecture, we'll learn all about the plant body in a simple and easy to understand way. So, let's begin. The plant body, plant organs. At first, many organs were recognized in a plant body, but later their number was reduced to 3, i.e. stem, leaf and root. The relationship of stem, leaf and root to each other and to the plant is still one of the basic problems of the plant morphology. It is unclear whether plant organs differ from each other essentially, or they are modification of one basic unit. However, evolutionary studies suggest that organization of oldest land plant was very resembling leafless and rootless Devonian plant, Rhynia. If seed plants have evolved from these plants, then the leaf, the stem, and the root would be closely related physiologically from an evolutionary point of view. Ontogenetically, the organs have common origin in the zygote and the resulting embryo, and in maturation, moments of the shoot, the leaf, and the stem, when fully grown, the leaf and stem merge with one another, internally and externally. The form, anatomy, function, and method of growth. Angiosperm's flower is considered homologous with a shoot and the floral parts with leaves. Both leaves and floral parts are thought to have originated from branch system. Development of plant body. A vascular plant starts its life as a unicellular zygote, 2N, which develops into an embryo and then into a mature sporophyte. This development includes division, enlargement, and differentiation. The physiological and morphological changes leading to specialization of cells, tissue, organ, or entire plant during the process of development from a meristematic state to mature one, of cells and organization of cells into complexes. The tissue systems of tissues. The embryo of seed plant has an axis bearing one or more cotyledons and tissues at a low level of differentiation. At the two opposite ends of the axis, meristems, tissue able to divide, of future shoot and root, called plumule and radicle, are present. After a certain period of vegetative growth, the plant enters reproductive phase. The increase in plant organs take place as a result of expansion in width and length. The resultant growth of root and vegetative and reproductive shoot is known as primary growth. The resultant plant body is termed as primary plant body, consisting of primary tissue. In thallophytes, algae, pteridophytes, and monocot, entire life cycle is completed in primary growth. In gymnosperms, some monocots and most dicots, increase in thickness of root and shoot take place by secondary growth. This growth results in the formation of secondary tissue. The secondary growth may be diffuse secondary growth, as it involves cells of ground tissue not localized in a specified region and found in palms, etc. Or it may be cambial secondary growth, when it is brought about by cambium, a lateral meristem, i.e. present away from the apex. The cambium producing secondary tissue, resulting in increase in diameter of cork cambium or phelogen, usually develop in peripheral regions, and periderm, a protective tissue which replaces the epidermis. End parenthesis. Organization of a vascular dicot plant. Internal organization. The units of a multicellular plant body, the cells are associated with one another in various ways, forming masses or tissues. The tissues arrange themselves into larger units, tissue systems. The body of a vascular plant is composed of 3 systems of tissues: the dermal, the vascular, and the fundamental or ground tissue system. The dermal tissue system provides outer protective covering of the plant and is represented by epidermis in primary plant body. During secondary growth, it may be replaced by periderm. The vascular tissue system is composed of 2 major conducting tissues, the phloem and the xylem. These tissues contain many types of cells, such as tracheids, vessel members, sieve tube members, companion cells, and parenchyma cells. The ground tissue system include parenchyma, basic composed of living cells which may differ in size, shape, and wall structure. Collenchyma, supporting tissue of young organs consisting of more or less elongated cells, the walls of which are usually unequally thickened, or into secretory structures. The ground tissue also contains mechanical tissue, sclerenchyma, the supporting tissue composed of fibers and sclerids. In stem, the vascular system occupies a position between epidermis and center of the axis. The in-between areas are occupied by some ground tissues, the cortex between epidermis and vascular region, and the pith in the center of the axis. In root, pith may be absent and the cortex is shed during secondary growth. In leaf, the vascular system consists of many interconnected strands embedded in the ground tissue. The mesophyll, photosynthetic parenchymatous tissue, situated between two epidermal layers of the leaf. The three tissue systems of primary plant body are derived from apical meristems. When derivatives of these meristems are partly differentiated, they may be classified as protoderm, procambium, and ground meristem, giving rise to epidermal, vascular, and ground tissue systems. Cell types and tissues. The following cell types and tissues are usually found in a vascular plant body. Epidermis. Epidermal cells form a continuous layer on the surface of primary plant body. The epidermal cells are of two types. A. Epidermal cells proper. They form main mass of epidermis, vary in shape, and often tubular. B. Guard cells, forming stomata. Epidermis may contain secretory and sclerenchyma cells. Epidermal cells of aerial parts of plant have a layer of cutin on the outer walls, which forms cuticle. Epidermis provides mechanical protection and restricts transpiration. During secondary growth, the epidermis is replaced by periderm in roots and stems. Periderm. It comprises of phellem or cork tissue, phellogen or cork cambium, and phelloderm. Phellogen arises in epidermis, cortex, the phloem, or the root pericycle, and produces phellem towards outside and phelloderm towards inside. Cork cells are usually tubular, without protoplast at maturity, and with suberized walls due to deposition of suberin. Parenchyma. Parenchyma cells form continuous tissues in the cortex of stem and root, and the leaf mesophyll. They also occur in vertical strands and rays in the vascular tissues. They are living cells, capable of growth and division, meristematic cells. The cells vary in shape, often polyhedral, having many sides, but may be stellate, star-shaped, or elongated. They are concerned with photosynthesis, storage, and wound healing. They may be specialized as secretory or excretory structures. Collenchyma. Collenchyma is a living tissue with unequally thickened walls and act as supporting tissue of young organs. The shape of cells varies from short prismatic to much elongated. Sclerenchyma. Sclerenchyma cells may form continuous masses, or they may occur in small groups or individually among other cells in any or all parts of the plant body. Sclerenchyma cells have thick, lignified, due to deposition of lignin, walls, and lack protoplast at maturity. They are strengthening elements of mature plant parts. There are two types of sclerenchyma cells. The sclerids vary in shape from polyhedral to elongated and often branched, and the fibers, generally LOM cells. Xylem. Xylem cells are found throughout the plant body, in association with phloem cells. The tissue is concerned with conduction of water, storage of food, and support. The main water-conducting cells are tracheids, and vessel members which join end to end into vessels. Storage occurs in parenchymatic cells, whereas the supporting cells are fibers and sclerids. Phloem. Like xylem cells, the phloem cells are present throughout the plant body, along with xylem. The tissue is meant for conduction of food, storage of food, and support. The chief conducting cells are sieve cells and sieve tube members, which join end to end to form sieve tubes and parenchymatous companion cells. Both cells are enucleate at maturity. The supporting cells are fibers and sclerids. Secretory structures. Secretory cells, the cells producing secretions, are present within other tissues as single cells or as groups of cells, and also as organized formations on the surface of the plants. For example, glandular epidermal cells, hairs, floral nectaries, etc. Internal secretory structures are secretory cells, intercellular cavities or canals lined with secretory cells, resin ducts, oil ducts. Section. Methods for studying anatomy of a plant. P. The principal method for studying the minute structure of the plant body is by means of thin sections of the plant material and maceration in which the individual cells are freed from one another. For adequate comprehension of the complex structure of most parts of the plants, study of sections cut in more than one plane is necessary. For the axis, a cylindrical structure, 3 planes, each at right angles to the other 2, are useful. One plane is transverse to the long axis, and the section cut out is termed as transverse section. The others are parallel with the long axis, i.e., longitudinal. Of the 2 longitudinal planes, that dividing the cylinder radially is the radial plane, and the resultant section is known as radial section, while that at right angles to the radial plane is the tangential plane, and the section cut out in this plane is called tangential section. Diagram showing transverse, radial, and tangential planes of the section. Let me know if you need help with anything else. If you found this video helpful, please give it a thumbs up, share it with your friends, and don't forget to subscribe for more easy biology lectures. Answer the following questions. 1. What is plant? 2. What are tissues? 3. What are fibers? If you know the answer of the following question, then write in comment section. If you have any question, then ask in comment section. Thanks for watching, and see you in the next class.